This video is about recombinant DNA technology. Over the past 30 years, tremendous advances have been made in the analysis of eukaryotic genomes. This progress began as molecular biologists learned to construct recombinant DNA molecules, which are molecules containing DNA sequences derived from more than one source. Recombinant DNA can be used in many ways. We will begin by considering one of the most important applications, the isolation from a genome of a particular segment of DNA that encodes a particular polypeptide. First, however, it is necessary to consider a class of enzymes whose discovery and use has made the formation of recombinant DNA molecules possible. There is restriction endonucleases. During the 1970s, it was found that bacteria contained nucleases that would recognize short nucleotide sequences within duplex DNA and cleave the DNA backbone at specific sites on both strands of the duplex. These enzymes are called type 2 restriction endonucleases or simply restriction enzymes. They were given this name because they function in bacteria to destroy viral DNA that might enter the cell, thereby restricting the growth of the virus. The bacterium protects its own DNA from nucleolytic attack by methylating the bases at susceptible sites, a chemical modification that blocks the action of the enzyme. Enzymes from several hundred different prokaryotic organisms have been isolated that together recognizes over 100 different nucleotide sequences. The sequences recognized by most of these enzymes are 4 to 6 nucleotides long and are characterized by a particular type of internal symmetry. Consider the particular sequence recognized by the enzyme EcoR1 as in the figure, as seen in this figure, as this segment of DNA is said to have twofold rotational symmetry because it can be rotated 180 degrees without change in base sequence. Thus, if one reads the sequence in the same direction 5' to 3' or 3' to 5' on either strand, the same order of bases is observed. A sequence with this type of symmetry is called palindrome. When this enzyme EcoR1 attacks this palindrome, it breaks each strand at the same site in the sequence, which is indicated by the arrows between the A and G residues. The red dots indicate the methylated bases in the sequence that protect the host DNA from enzymatic attack. Some restriction enzymes cleave bonds directly opposite one another on the two strands, reducing blunt ends, whereas others, such as EcoR1, make staggered cuts. The discovery and purification of restriction enzymes have been invaluable in the advances made by molecular biologists in recent years. Since a particular sequence of 4 or 6 nucleotides occur quite frequently simply by chance, any type of DNA is susceptible to fragmentation by these enzymes. The use of restriction enzymes allows the DNA of a human genome or that of any other organism to be dissected into a precisely defined set of specific fragments. Once the DNA from a particular individual is digested with one of these enzymes, the fragments generated can be fractioned on the basis of length by gel electrophoresis. Different enzymes cleave the same preparation of DNA into different sets of fragments, and the sites within the genome that are cleaved by various enzymes can be identified and ordered into a restriction map such that depicted in this figure. When we look at this figure, the construction of a restriction map of the single circular genome of DNA tumor virus polyoma. Autoradiographs of 32p labeled DNA fragments that have been subjected to gel electrophoresis. The gel on the left shows the pattern of DNA fragments obtained after a complete digestion of the polyoma genome with the enzyme HPA2. To determine how these 8 fragments are pieced together to make up the intact genome, it is necessary to treat the DNA in such a way that overlapping fragments are generated. Overlapping fragments can be produced by treating the intact genome with the second enzyme that cleaves the molecule at different sites or by treating the genome with the same enzyme under conditions where the DNA is not fully digested as it was in the left gel. The two gels in the right represent examples of partial digests of polyoma genome with HPA2. The middle gel shows the fragments created by partial digestion of this superhelical circular DNA. And the gel on the right shows the HPA2 fragments found after the circular genome is converted to a linear molecule by EcoR1, an enzyme that makes only one cut in the circle. The restriction map of the linearized polyoma genome based on cleavage by HPA2, that is something which is represented by B. The 8 fragments from the complete digest are shown along the DNA at the top. The overlapping fragments from the partial digest are shown in their ordered arrangements below the map. So carefully observe this picture. You could see origin and some other regions which we explained with the help of a text. Next, formation of recombinant DNAs. Recombinant DNAs can be formed in a variety of ways in the method as shown in this figure. DNA molecules from two different sources are treated with a restriction enzyme that makes staggered cuts in the DNA duplex. Staggered cuts leave short single-stranded tails that act as sticky ends that can bind to a complementary single-stranded tail on another DNA molecule to restore a double-stranded molecule. In the example depicted in this figure, one of the DNA fragments that will make up the recombinant molecule is a bacterial plasmid. Plasmids are small circular double-stranded DNA molecules that are separable from male bacterial chromosome. The other DNA fragment in the figure is obtained from human cells following treatment with the same restriction enzyme used to open the plasmid. When the human DNA fragments and the treated plasmids are incubated together in the presence of DNA ligase, the two types of DNA become hydrogen bonded to one another by their sticky ends and then ligated to form circular DNA recombinants as in this figure. The first recombinant DNA molecule were formed by this basic method in 1972 by Paul Berg, Herbert Boyer, Annie Klang, and Stanley Cohen of Stanford University and the University of California, San Francisco, making the birth of modern genetic engineering. By following the procedure just described, a large number of different recombinant molecules are produced, each of which contains a bacterial plasmid with a segment of human DNA incorporated into its circular structure. Suppose you were interested in isolating a single gene from a human genome, for example, the gene that codes for insulin. Because your goal is to obtain a purified preparation of the one type of recombinant DNA that contains the insulin coding fragment, you must prepare this one fragment from all of the others. This is done by a process called DNA cloning. When we look at the structure, you could see bacterial plasmid, human chromosome. Same restriction enzyme splits both types of DNA. Human DNA fragment joins with plasmid by sticky ends and nicks are sealed by enzyme. So this is a formation of recombinant DNA molecule in this example. A preparation of bacterial plasmid is treated with a restriction enzyme that makes a single cut within each bacterial plasmid. The same restriction enzyme is used to fragment a preparation of human genomic DNA into small fragments. Because they have been treated with the same restriction enzyme, the cleaved plasmid DNA and the human DNA fragments will have sticky ends. When these two populations are incubated together, the two DNA molecules become non-covalently joined to each other and they are then covalently sealed by DNA ligases, forming a recombinant DNA molecule. Thank you for listening.